Myself, Dr. M. Gangaraju. I am working as a professor and head Department of Pharmacology, Gogaraju Rangaraju College of Pharmacy, Hyderabad. And for this session, my talk is on importance of receptors in cell signaling. The contents to be discussed are introduction, receptor, and we need to find out various types of receptors like G protein coupled receptors, tyrosine kinase receptors, and ligand gated ion channel receptors, their importance in cell signaling, and the secondary messengers, summary, and finally the references. Coming to the introduction, we basically should know what is a receptor. So I'll define the receptor as a protein molecule that receives the chemical signals from outside a cell. And these receptors, which are acting as the proteins, they are always located on the surface of the cells. And as we at this particular juncture, we need to know the importance of receptor in cell signaling. It is also mandatory that we need to know what is a cell signaling. Cell signaling is either mechanical or biochemical and can occur locally or for long distances. Coming to the receptor is a protein which receives a chemical signal from outside. As depicted in this particular picture, the ligand combines with a receptor, and this coupling will try to take part in which gives you the outcome of cell signaling. Ligand and receptor always follows the lock and key principle. And what happens to a receptor when a ligand binds to it? Molecules like drugs, hormones, or neurotransmitters that bind to a receptor are known as ligands. The binding can be specific and reversible. A ligand may activate or inactivate a receptor. But remember that activation may increase or decrease a particular cell function. Each ligand may interact with multiple receptor subtypes. Coming to the importance of G protein coupled receptors, G protein coupled receptors are the largest and the most diverse group of membrane receptors in eukaryotes. In the inactive state, the G protein binds to a nucleotide GDP, guanosine diphosphate, and the G protein has three subunits, namely alpha, beta, and gamma. Activation of the receptor by the neurotransmitter like dopamine causes the alpha subunit to exchange its GDP with GTP. The protein thus has two functional formations, and the best examples for GPCR are adrenergic, histaminic receptors, muscarinic receptors, dopamine receptors, etc. Next, shifting over to the next type of receptors playing a major role in cell signaling are tyrosine kinase. Basically, tyrosine kinase is an enzyme, and these are high affinity cell surface receptors for many polypeptides, growth factors such as cytokines and hormones. These receptors differ from other cell surface receptors in that they contain an intrinsic enzyme activity, that is the role played by the tyrosine kinase receptors. In particular, the binding of a signaling molecule with an tyrosine kinase receptor, it activates the enzyme tyrosine kinase in the cytoplasmic tail of the receptor. And insulin receptor is the best example which comes under the tyrosine kinase receptors. Next, this slide will show you the phosphorylation cascade. Initially, you can easily make out a relay molecule activates the protein kinase 1. Once the protein kinase 1 is activated, it transfers a phosphate from ATP to an inactive molecule of protein kinase 2, thus activating this second kinase. Once the protein kinase 2 is activated, then it catalyzes the phosphorylation of protein kinase 3. And finally, the active protein kinase 3 phosphorylates a protein that brings about the cellular or the cell response to the signal. And finally, the outcome of this is a cellular response. Remember that the enzymes known as protein phosphatases PP They catalyze the removal of phosphate groups from the proteins, making them inactive and available for reuse. This is the way how a phosphorylation cascade proceeds in cell signaling. Coming to the third type of receptors, that is ligand-gated ion channel receptor. The title itself or the heading itself will tell that they have an effect on the ion channels. As we already know that ion channels are located on the surface of the plasma membrane of most of the cells here. Once the signaling molecule, which is known as the ligand, occupies its own receptor and try to act on the channels, automatically the gates of these channels will open, letting out or letting in the various molecules, and finally the outcome of this is the cellular response. And within a few milliseconds itself, once the gates might also be closed. The best examples for ligand-gated ion channel receptors are GABA receptors and the nicotinic receptors. Now, something we'd like to talk about the transduction. Transduction plays a major role in cell signaling. As we already know that we have three important stages in cell signaling. The first one is the reception, second one is the transduction, and finally the third one is the response. So here in this particular slide, we'll give you the outcome, how a cellular response is produced. Initially, we can easily identify in the picture itself the signaling molecule which is acting as the first messenger try to couple with the G protein, and this combination in the presence of GTP, it acts on in the presence of phospholipase C, will try to form a compound known as PIP2, phosphoinositol diphosphate. Once PIP2 is formed, the outcome of PIP2 will be two second messengers. One is the DAG, diacylglycerol, and the second one is the inositol triphosphate. These two are known as second messengers or secondary messengers. Once these two are formed, DAG will be utilized for other reactions, but IP3 will try to act on the IP3 gated calcium channels which are located on the plasma membrane of the endoplasmic reticulum. Once these IP3 gated calcium channels are activated, the gates will be opened. The calcium from the endoplasmic reticulum will be released, which automatically increases the level of cytosol calcium levels. Once calcium is released, it will be utilized by various proteins. They will become activated. Once these proteins are activated, the outcome is the cellular response. Now coming to the importance of second messengers and cyclic AMP. In the picture itself, we can easily make out the first messenger, which is known as the signal molecule such as adrenaline or epinephrine or some other molecule. They'll try to couple with the G protein, and once they couple, G protein in the presence of GTP, we can make out the formation of cyclic AMP, or ATP in the presence of adenyl cyclase gets converted into cyclic AMP. Once cyclic AMP is formed, the protein kinase A will be activated, and if this is activated, the outcome is a cellular response. Now this is a slide which gives you different aspects like what is ATP, cyclic AMP, and AMP. So you easily can make out adenosine triphosphate in the presence of an enzyme, adenyl cyclase. The product formed is a cyclic AMP, and this cyclic AMP in the presence of phosphodiesterase gets converted into adenosine monophosphate. Second messengers, the calcium. Already this calcium, mostly in cells, it is concentrated in two regions. One is the mitochondria and other one is endoplasmic reticulum. These two structures are the storage sites of calcium. When the cytosol calcium levels has come down, immediately due to a series of responses, the calcium pumps gates will be opened, and automatically the calcium will be released into the cytosol. Calcium ions, its importance it is a second messenger. Its importance, we'll try to study its importance in cell signaling. Initially, a signal molecule binds to a receptor leading to the activation of an enzyme phospholipase C. Once this enzyme is activated, it cleaves a plasma membrane phospholipid called PIP2 into DAG and IP3. DAG functions as the second messenger in another pathways, but IP3 inositol triphosphate will try to quickly diffuses through the cytosol and binds to an IP3 gated calcium channel in the endoplasmic reticular membrane, causing it to open. Once calcium ion flow out of the ER, that is down their concentration gradient, raising the calcium level in the cytosol. The calcium ions activate the next protein in one or more signaling pathway. Once the proteins are activated, the outcome is the cellular response. This is the role played by the calcium as a second messenger in cell signaling. Coming to the summary of this particular topic, receptors are protein molecules. They receive the chemical signals from outside a cell. Various receptors like G protein coupled receptors, kinase linked receptors, and ligand gated ion channels play an important role. Second messengers are intracellular signaling molecules released by the cell in response to exposure to extracellular signaling molecules. Examples of second messenger molecules include cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol, and calcium. Thank you.